Hi, my name is Sarah.
I'm Elaine.
And we're scientists at UW-Madison in the genetics laboratory. In our lab, we study how different modifications on DNA affect how the cell interprets that DNA. And today, we're going to walk you through a short, fun activity where you can extract DNA from a strawberry using some household items. And so our first step, and things we're going to need a plastic bag, some water, 2 containers for 2 people so that we can filter our strawberry through, salt, dish soap, isopropyl alcohol,
Some
measuring spoons, cheesecloth, and funnels, and some wooden sticks to analyze our DNA later. So the first step, we're going to take our strawberries. We've got kind of small ones here, so I'm going to take 3 strawberries and I'm going to remove the green part, so, and I'm going to put them in our gallon-sized bags. And then I'm going to get all the air out of the bag, have only the strawberry filling out the bag, like so. And then I'm going to crush the strawberries with my hands.
So how do you normally crush plant tissues as a scientist?
As a scientist, I usually use liquid nitrogen to crush up my plant cells. And I do that because I want everything to stay ultra cold as I'm working with it. I, as a scientist, do not just extract DNA, but I extract proteins and RNA as well. Which are molecules that have to be really cold to be able for me to analyze them.
That's super exciting.
Yeah. But for DNA, since it is very stable, we can crush it at room temperature and it will be totally fine, like we are doing now with the strawberry. See, so this process is a mechanical breaking down of the material. We are smushing everything to try to break down the cell membranes and the cell walls so that we can extract the DNA that's inside the nucleus of our cells. How's your crushing going?
Sounds like a pretty good jam.
Yeah, it's kind of like making jam. It's the first step.
Yeah.
Except for what we're going to do next you're not going to want to have to eat it.
And we add salt instead of sugar. In our lab, we also have something called a genome blender, where you could shake the plant materials at a certain frequency for a certain time, which is super convenient to use.
We're often working with a lot of plants at once, and so doing something like this is a little tedious. So we want to try to use something like she's talking about to grind a bunch of samples at once without mixing them up. And so once your solution looks somewhat like this, nice and smushed up, we're going to make our extraction buffer. And so I'm going to make extraction buffer for 2 extractions, since there's 2 of us here. But if you're only doing it for 1, just use 1 of each. So first, what we're going to do is we're going to measure out 1/3 cup of water. And so just any water from the tap will work. And so the water acts as a solvent, or the base of our extraction buffer that will allow us to get the DNA out of the strawberries. So next we want to add dish soap. Any dish soap will work. And what this does is it's a soapy texture that will break down the membranes that make together the cell membrane and the nuclear membrane of the plant. And this is what the membranes are, what protects the cellular components of the strawberry, and the soap helps us break those down. And then our final ingredient is salt. Anyone know what salt's going to do in this? Do you know?
I'm gonna think about it.
So DNA is negatively charged, and so salt helps it attract the negatively charged particles out of the strawberry into a clear solution. And so we're going to add 1/2 teaspoon of salt per extraction. 1/2 and 2 halves. And then we're going to give this a really good mix so it's all dissolved. And it should dissolve quite fast because salt dissolves in water very well. So you should have a nice soapy mixture. And then we are going to add 3 tablespoons worth of our extraction mixture to each of our strawberry mixtures.
I hope it doesn't take close to
2 and 3. Now this is not something you want to eat now. You want to measure your own?
Here.
And then this step can get a little messy. So after you add your mixture to your strawberries, you want to seal it up and make sure you get all the air bubbles out. And then give it a good squish before. And start to look and see how your mixture is changing. How does it look different? What do you think the soap and the salt are doing? Right? Look at how the texture changes and how the soap changes it. You're gonna give it a good mix for a couple minutes.
So what do you normally use in the place of the dish soap?
In dish soap, we've got some lab grade detergents that we'll use instead. Something like chemicals called MP40 or Triton can be used in place of dish soap in a lab setting. But for our purposes, you know, Dawn dish soap or any type of dish soap works really well. And so you should have a really nice and soapy strawberry mixture. It looks pretty gross. But what we're going to do next is we're going to filter it through some cheesecloth to catch all the big pieces. And so we're going to use the cheesecloth that's filtered and then put through the funnel so we don't make a mess. I'm doing mine in just a normal jar that you could find in your house, but Elaine is going to be doing hers in a graduated cylinder so we can get a better look at what the isopropyl alcohol is going to do for the DNA later. So you're just going to line your funnel with some cheesecloth as such. And then we're going to very carefully, try not to make a mess, pour our strawberry mixture through the cheesecloth into the funnel. And so it will take a little bit of time for everything to filter through. So while these filter, what got you into science, Elaine? Why did you want to be in genetics?
I guess I was originally an undergrad in botany, and I just feel that genetics and epigenetics are really important parts of plant science and how to learn about like life in general, why they are plants, why they look that way, why they're green and all that. It's all very intricately monitored through genetic code and epigenetics, which is super interesting. How about you?
I agree. Yeah, I am in graduate school right now, and so when I was younger, I was always really interested in how the world works, especially plants. I thought they were especially beautiful, and it was always really interesting to me how resilient they were and how they can face any sort of environmental stress and still deal with it. Unlike animals and humans, they can't just walk away when things are bad. They have to deal with the stress that they face at this point in time. And so I decided I wanted to understand how they handle these stresses at the genetic level, since it's the basis of the organism. It's the blueprint, and it gives it the guidance for how they're going to change in response to environmental stress. And so epigenetics, which is something that we focus on, the way to study these different stresses, or these different responses to stress. So that's how I got through. It may take some time for all your juice to start filtering out of the funnel and the cheesecloth. But just be patient. It's important that we get a really pure sample and we don't have any of the larger pieces.
What are some normal ways you analyze epigenetics components after you extract the DNA?
That's a good question. So what we use with the DNA afterwards? I will oftentimes look for if there are certain sequences present, using different techniques such as PCR, or if I want to know if the number of these sequences are present, I will use a more quantitative way to measure these things, such as qPCR, or we use sequencing methods to be able to analyze on the whole genome scale how the sequences of DNA change under different stresses or treatments. And so I think we have enough. So we're going to move our cheesecloth and our funnels out of the way, and then we're going to very gently, and I think
It smells good.
it's nice and soapy and strawberry. We're going to try this on Elaine's because it looks the best, but we're going to very, very gently take our isopropyl alcohol and very slowly pour it on top of our mixture. And so as you can see, as you do this, do you see something in the middle there? What do you think that is? That is the strawberry's DNA itself. And so what we could do with our skewer, maybe I need to add a little bit more. Our skewer is there, it's very pronounced. You see that white clumpy thing in the middle? That is the DNA. So what we do is fish some of it out. So you've probably never seen what DNA looks like in person, but here it is. It's this globby white thing that came out of the strawberry. And so why it looks so white and gloopy is because it has all the proteins that are involved in the DNA still present and wrapped around it. And these proteins are really important for the integrity and the organization of the DNA within the nucleus. So try this at home. Let us know if it works and what you would like to do with strawberry DNA when you extracted it. Thank you. Bye-bye.